Thank you very much for inviting me to give this talk on the role of CAR T cells in myeloid malignancies. And I've pitched this talk as lessons learned from lymphoid malignancies. I do have some disclosures: research funding from Novartis, Charisma, and Interius Therapeutics, stock and ownership from Charisma and Interius, consulting from Intellia, and contracted research with Hemogenics and Novartis. The therapeutic landscape of CAR T cells for hematologic malignancies is illustrated here in this very nice heat map from a recent publication. On the x-axis, on the horizontal there, we see a variety of different target antigens that are being investigated in clinical trials. And on the y-axis, we see a variety of different hematologic malignancies. And MDS and myeloid leukemia are the top two rows. And the heat color scheme represents the number of clinical trials that are being done in that indication and using that particular target antigen. So, for example, you could see that MDS is generally relatively poorly represented amongst the CAR T-cell clinical trial spectrum at the moment, and myeloid leukemia is predominantly accounted for by clinical trials targeting CD123 or CD33. So the currently recruiting trials, at least for a few months ago that we put together for a publication, include, generally speaking, trials that target CD123 or CD33, sometimes CLL1, also known as CLEC12A, and particularly in the setting of patients who have MDS or myeloproliferative neoplasms, there's a handful of other potential targets, including IL-1 RAP and NKG2D, as you can see at the bottom of the slide. The complete response rates in hematologic malignancies, specifically lymphoid malignancies, are illustrated on this slide. And one of the most gratifying things about working in this area, I think, is the reproducibility of these results. So across different types of patients, different countries, different centers, or different companies, different products, we see about the same thing, which is patients with ALL have a very high complete response rate of 80 to 90%, and those are very deep responses, MRD negative very often. Non-Hodgkin lymphomas, typically 50-ish percent complete response rates, and CLL is lower. This is with the IWCLL response rate, though, criteria, though, so perhaps a little bit tough with respect to achieving a complete response on those. The converse is in multiple myeloma, where the VGPR and CR rates are reported together, and so probably a little generous when it comes to myeloma response rates. The key, though, is despite the fact that AML is not an uncommon disease, certainly not for the people in the audience here, I was able to find a total of 46 patients who were reported in the literature on case reports or small case series as receiving CAR T cells for AML, and of whom 7 out of the 46 achieved what I manually curated as deep response. So having looked deeply into the papers and worked out that those patients had the sort of desirable deep response. So that's 7 out of 46 in the whole world. In 9 different and small trials. And I think the difference there really speaks for itself with respect to the comparison with lymphoid malignancies. So what are the challenges for us? I think of these as target selection that speaks to toxicity and efficacy, efficacy in itself, and then logistics and feasibility. One thing that we've learned from our friends in the B-cell field is that B-cell aplasia is a surrogate for CAR T-cell functional persistence. So here are 2 patients from one of my trials. This is a CAR T-cell plus ibrutinib trial for CLL. Patient number 1 is on the left, patient number 5 is on the right, and I've superimposed the B-cell reduction or B-cell quantification in the blood in purple on top of the CAR T-cell expansion by flow cytometry in cyan. Patient number 1 shows the presence of B cells, which go down when the CAR T cells expand, and then the CAR T cells go away at around day 28 and the B cells return. And that's not, of course, not what we want. On the right side is patient number 5, who achieved exactly what we want, which is a MRD negative remission, so complete reduction of the B cells in the blood and also in the bone marrow as the CAR T cells go up. And then the CAR T cells persist for at least 12 months. And in fact, we have patients who have persistence of the CAR T cells for years. And that goes along with absence of B cells for years. And while that is entirely feasible in when you're dealing with B cells, it is not feasible when you're dealing with myeloid cells, because we need those myeloid cells. So having said all that, the main targets still are lineage antigens. And so CD19 or 22 for B cells, BCMA for myeloma and plasma cells. In AML, here are some of the leading antigens. You can see that CD33 is expressed at high levels on leukemia, bulk leukemia cells in red and leukemia stem cells in dark brown, but also at very high levels on some of your progenitors, particularly your myeloid progenitors, and particularly once you start to differentiate into monocytes and neutrophils, very high levels of CD33, suggesting that there would be expected to be myeloid toxicity. The case is identical in the setting of CD123, which also has been advanced as a potential therapeutic target. CLL1 or CLEC12A is really not particularly highly expressed at all on putative leukemia stem cells, that is immunophenotypically defined stem cells, and is also very highly expressed on neutrophils, granulocytes, and monocytes. So, in my opinion, not a terribly good target there. So, another way of thinking of this is asking ourselves the question, what are we trying to achieve? And when I talk to my patients about immunotherapy or indeed any other therapy for AML or MDS, I try to explain in terms that people with general level of experience will understand, and the ability to definitively differentiate crops from weeds, leukemia cells from hemopoietic stem cells is critical when it comes to the therapeutic window. So what do we think we know? Well, we think we know some of the potential surface antigens on malignant myeloid cells in the bone marrow. But those antigens are also all present on normal cells as well. And so we really are at the point where it is incredibly difficult, and I would say impossible, to differentiate malignant cells from normal cells with a single and possibly even with combinatorial targeting. But what we do know is that there are disease-associated and disease-specific and disease-driving mutations associated with myeloid malignancies, and those are generally, with some exceptions, not present on normal cells. So can we put those two bits of knowledge together and move perhaps our understanding of the field forward? I'll show you one or two slides from a technology that I think is quite interesting and uses single-cell DNA sequencing, not RNA sequencing, but single-cell DNA sequencing, targeted amplicon sequencing, along with sequencing essentially of antibodies. So you can link antibodies to fluorochromes, and that's how you do flow cytometry. But you can also link antibodies to oligonucleotides, and then sequence them. And by separating individual cells from patients' bone marrow in droplets, you can do single cell sequencing and find out both the pattern of protein expression on the surface of the cells, like flow cytometry, as well as the pattern of mutations within the cells. And that allows you, in my mind at least, the most direct genotype-phenotype correlation. So this has been published recently by other groups, notably by Ross Levine's group and another group at MD Anderson. Very, very nice work. We've done some of this work as well. And here, this is a primary patient bone marrow sample where we were able to identify 5 distinct clones, as well as potentially residual wild-type normal cells. I call them normal cells because we could not detect any mutations, but of course there could have been mutations that we didn't detect or didn't look for. And we were able to do this at a single-cell resolution, as I mentioned. And then the sort of block there in the middle of the top of the slide, if you can see my cursor, shows us the different clones, and the size of the clone is defined by the number of cells or the percentage of cells in the sample that we assign to that clone based on the presence of these mutations. And you can see that the single most abundant clone was clone number 3, which is the one that had a DNMT3A mutation and then 2 different NPM1 mutations. And we had some residual, perhaps wild-type or normal cells, but very few. And then the key of this slide here is really looking at the protein expression on the surface of these cells as defined as normal or abnormal or belonging to different clones. And for those of you who are not used to looking at such things, we are looking at increased expression of each target antigen going from left to right. So on the left of each violin plot is the lowest expression of the antigen, in this instance CD33, and then that sort of the bulge in the middle is the highest numbers of cells, the median number of cells that express in the clone, and you can see that most cells express CD33, and the level of expression of CD3 is about the same across the different clones, and that is also true for the other antigens such as CD34, CD123, CD135, which is FLT3. And in fact, if you look at CD135, you can see that the expression of FLT3 is higher on our normal cells than it is on any of the clones. And another way of showing the same data is a sort of pseudo flow cytometry plot one by one. Here is CD34 versus CD33, or CD34 versus CD38, or CD34 versus any of the other markers. And here we're superimposing clonal identity over each and every single cell as represented by dots in this plot. And you can, I think, pretty clearly see that there is really no difference in any of the clones or the putative wild-type clone with respect to their cell surface antigen expression. And that's really important when it comes to thinking about the potential ability to design leukemia-specific or MDS-specific immunotherapy. So one way of getting around this was published by my group a couple of years ago. In this instance, we decided to engineer normal stem cells to lack the shared antigen. And I showed you data on shared antigens just a second ago. So rather than trying to find a specific antigen on the leukemia cells or malignant cells, we decided to create de novo a cancer-specific antigen by editing using CRISPR the normal cells, donor cells. And by doing so, we hypothesized that we would be able to treat AML or MDS with full strength with our CAR T cells against CD33, but that would eliminate disease, but that it would leave unharmed normal cells that were edited to lack CD33. And this was published, so I won't spend too much time going through the results, but suffice it to say, I think that we're able to achieve very high level knockout of CD33 in normal human CD34 positive progenitor cells. They look the same as each other except for lack of CD33. We engrafted cohorts of mice with CD33 deficient human stem cells or with a control guide, and found that we indeed had long-term loss of CD33 in our hemopoietic system, yet that did not result in skewing. We still had plenty and equivalent numbers of granulocytes and monocytes, CD11B and CD14 cells respectively. And when we looked at them in the bone marrow, there was no difference in the progenitors or stem cells of mice, whether they received human cells lacking or containing CD33. And then we pushed the system one step further by engrafting such mice with a leukemia cell line called MUL14 that was expressed luciferase, and we could then detect leukemia engrafting in these mice. And in so doing, we then could treat the mice with CAR T cells against CD33. And we show here on the slide that the leukemia was able to be eradicated by the CAR T cells as expected. But the key is really in this slide, which is having eradicated disease, do we have preservation of hemopoiesis in mice that received CD33 deficient human stem cells? And the answer is yes. You can see in panel H that we have preserved monocytes in the blood, only in the blood of the mice that were CD33 knockout recipients, and preservation of the early hemopoiesis in the bone marrow in panel I. And this we also validated in non-human primates by a collaboration with Cindy Dunbar at the NHLBI, and we are moving towards a clinical trial of this exact concept in humans. And I would add that this was independently verified and reproduced by two other groups subsequently. So the way this might look would be a allogeneic stem cell transplant into a patient with, say, relapse refractory AML or high AML that is at high risk for relapse, who is first treated with a allogeneic stem cell transplant with an engineered graft, where the graft is engineered to lack human CD33. And upon engraftment, the patient is then treated with an infusion of allo CART33 cells in order to attack their malignant hemopoiesis, but leave their normal hemopoiesis. So in the last few minutes, I want to share with you a few interesting vignettes that are of great relevance to particularly MDS, and MPNs, I think. And I'll explain why in a second. So the first case was a patient that I treated with anti-CD123 CAR T cells for acute myeloid leukemia. This is unpublished as yet. This is a patient who had pre-existing myelofibrosis, which was JAK2 positive for many years. And eventually, this patient progressed to acute myeloid leukemia, and we treated her in March of this year with CART123. She developed grade 2 cytokine release syndrome, which resolved, and she went home feeling very well. But interestingly and very uncharacteristically, over the next couple of weeks, she developed progressive and very severe pancytopenia down to a neutrophil count of 0. Yet without fever and without obvious CRS. And then about 3 weeks after infusion, she was admitted with a fever and hypotension and thought really to have febrile neutropenia, perhaps gram-negative sepsis in the setting of neutrophil count of 0. We treated her for this. We also added tocilizumab just in cases of cytokine release syndrome, but unfortunately within about 18 hours, she unfortunately progressed to irreversible multi-organ failure in the setting of a progressive and profound lactic acidosis, type B lactic acidosis, and severe hypoglycemia. Subsequently, we found out that her CAR T cells expanded at kinetics that were really unusual and continued to expand over time. This was not cytokine release syndrome. We looked at the cytokines in the blood, and they were actually falling at that time. We were able to get an autopsy which showed CAR T cells everywhere, but presumably in the blood, because there was no evidence of tissue infiltration or tissue damage by the CAR T cells, which led us to hypothesize that perhaps this patient with a long-standing stem cell mutation, that is a JAK2 mutation in her stem cells, had at some point in the last 20, 30 years produced some T cells with the JAK2 mutation. And that if that was the case, those T cells might have a unique phenotype that had not been observed before. So using single-cell RNA sequencing, we looked at the blood of actually other patients, not this patient yet, but found that we can find the CAR, we can find T cells, B cells, and monocytes, and we can actually find JAK2 as well as the downstream STAT3 and STAT5A and STAT5B, as well as the upstream receptors in all of those cells, including T cells. And so we then wondered whether, indeed, in this patient that I described to you, whether there was actually JAK2 mutated in the T cells. And we hypothesized that it may not be detectable in the infusion product because there would be so few cells at that time, but it would be enriched over time. And that's in fact what we found. So here I show you the patient's blood looking at the JAK2 V617F allele frequency on the left and the CAR T cell quantification on the right, and then days peri-infusion on the bottom. And you can see that while there's certainly JAK2 in the marrow at baseline, the infusion product contains no JAK2 mutation. And as we increase our CAR T cell numbers over time, and eventually at the latest time points where we have no myeloid cells in the blood, we actually have quite a few JAK2 V617F, at least a allele frequency thereof, suggesting, but not proving as yet, that this is in fact the case. And we had made inadvertently JAK2 V617F co-stimulated CAR T cells. Two more patients to illustrate the point. I told you about JAK2 in MPNs. A colleague of mine, Joe Frieda, published in Nature a few years ago the outcomes of a patient with CLL who had a TET2 mutation, perhaps CHIP underlying all this, and who had a remarkable response, essentially deriving from a single cell that was a CAR-positive T cell directed against CD19 in CLL, and that single cell eradicated that patient's disease. You can see here on the right side of the slide, all the CAR T cells at post-infusion belong to a single TCR family. Using TCR sequencing, we're actually able to find that was really a single clone, and that's in panel C below. And then the panel D on the bottom right really illustrates to you the exceptional expansion of those cells. And then finally, work done by Joe showed how this happened. Turns out that the CAR integrated into the TET2 locus in a single allele, but the other allele was already mutated in this patient with, as I said, presumably underlying CHIP, and that gave the T cells again conferred upon them a unique phenotype. So to finish off, I want to present one patient who represents what we should expect, I think, to see in myeloid-specific CAR T cells. This is a 55-year-old male with AML that relapsed after a prior sibling allotransplant and was resistant to all therapies subsequently. At baseline, on October 8th of last year, had 75% marrow blasts, was treated with CAR T cells, and then achieved 0% blasts, and in fact was completely aplastic for many months with a molecular MRD positive CR, nonetheless, at least no evidence of disease. And as expected, perhaps with months of aplasia, this patient developed an invasive fungal infection such that we spent quite a lot of time trying to treat that with antifungals and granulocyte transfusions. And eventually we gave this patient a purified CD34 infusion to reconstitute his hemopoiesis, which occurred beautifully, normal counts, and eradication of his disease with MRD negativity and a good outcome so far. So to sum up, the way I see things for myeloid malignancies and CAR T cells, I would say that we're at the end of the beginning. We know the platform works in lymphoid malignancies, and we've learned and are learning from lymphoid malignancies with respect to what to expect in myeloid malignancies. AML responds to some immunotherapy, but not others. There are early indicators of response to CAR T cells, as I showed you, and also to BiTEs, as has been recently published. The things we need to think about are target antigen selection, toxicity, on-target and off-tumor toxicity, efficacy, and rational combinations, which I've listed a few potential ones on the right side of the slide. We can think about combining with checkpoint inhibitors. How do we deal with immunosuppressive metabolites elaborated by malignant cells and the other immunosuppressive cells in the microenvironment? So thank you very much for your time, and I acknowledge my lab and collaborators and funding sources for this work. Thank you.